Hello, this is Professor Nikitakis from University of Athens, Greece. It is my honor to be a speaker in this great event, and for that I would like to thank the organizing committee and especially the chair, Professor Riza Chaisuparat, my dear friend and colleague. The topic of my talk will be oral potentially malignant disorders, predicting and preventing transformation to oral cancer. This is the outline of my lecture. After a short introduction, we will define the problem of malignant transformation, and we will briefly describe the basic histopathologic and molecular changes and markers in oral PMDs. Then we will proceed with a discussion on chemoprevention and its role, and we will briefly present some of our studies as an example of the long road from bench to bedside applications of oral PMD research. Finally, we will conclude summarizing the limitations and challenges we face, as well as the potential and hopes in this field. Let's start with a short introduction on oral potentially malignant disorders. The term potentially malignant disorders was introduced by WHO in 2005 to encompass the previously used terms premalignant lesion and premalignant condition. The latter terms are still commonly used nowadays. Although the definitions have changed over the years, they all emphasize that the terms premalignant or potentially malignant indicate an increased risk of cancer development compared to the corresponding normal state. Here we see the major classification of oral potentially malignant disorders. The most frequent ones are leukoplakia and erythroplakia. There are many others less frequent, such as lichen planus, the malignant potential of which has been debated over the years, actinic cheilitis, some lesions in reverse smokers, as well as submucous fibrosis, while there are also other more rare conditions. In a paper just published by the WHO Collaborating Center for Oral Cancer, it was suggested that the list of oral potentially malignant disorders should also include proliferative verrucous leukoplakia as a distinct entity, as well as oral lichenoid lesions and oral graft versus host disease based on sufficient evidence to justify their premalignant potential. On the other hand, it was proposed to remove epidermolysis bullosa from the list due to limited epidemiologic evidence of its premalignant potential. Several definitions of oral leukoplakia have been proposed in the past. As an example, this definition by WHO says that oral leukoplakia is a white plaque of questionable risk, having excluded other known diseases or disorders that carry no increased risk for cancer. The common theme in the various definitions is that leukoplakia is a diagnosis of exclusion. It remains a clinical term that does not indicate specific histopathologic features, so that the presence of dysplasia is not a prerequisite for the diagnosis. At this point, please allow me to show you the cover page of the special issue published by Oral Surgery, Oral Medicine, Oral Pathology, Oral Radiology in June 2018, which was devoted to oral PMDs, or otherwise called potentially premalignant oral epithelial lesions. I had the privilege to be the guest editor of this special issue and to coordinate a team of amazing authors, including some of the world leaders in the field, as you can see. I would welcome you all to visit this special issue, which is available online free of charge for a comprehensive review and update on oral PMDs. Of course, the major problem in oral PMDs and what we strive to avoid is their malignant transformation. Let us start by defining the problem, which is to accurately determine the likelihood of malignant transformation of an oral PMD. Why is this important? Because it will help us decide what the most appropriate course of action is based on how dangerous a given lesion is. And it is generally accepted that clinical and histopathologic parameters alone do not accurately predict the risk of malignant transformation. In this table from the review paper by Spite and colleagues, we can see that the rate of malignant transformation varies from study to study, but it is overall quite significant. And here are some examples of malignant transformation of pre-existing oral potentially malignant disorders. And here again. This table summarizes the major clinical and histologic features that have been associated with an increased risk of malignant transformation, including a large size of the lesion, a non-homogeneous texture, the red color, the localization in the tongue or the floor of the mouth, the female gender, an age above 50, as well as patients who are non-smokers, while the histologic features include severe dysplasia, presence of high-risk HPV subtypes, aneuploidy, and loss of heterozygosity. And again, the same features presented as a practical algorithm. Let's take a closer look at the histopathologic changes and their potential as markers in oral potentially malignant disorders. Oral PMDs may or may not harbor dysplastic changes that range from mild to severe to even carcinoma in situ. In this review paper by Müller, we can see nice illustrations of some of the well-known features of oral epithelial dysplasia, such as atypical mitotic figures, pleomorphism, and large hyperchromatic nuclei with multiple nucleoli. Furthermore, architectural changes can be discerned, such as loss of polarity and cohesion, and premature keratinization. Here is a summary of the architectural and cytologic features of oral epithelial dysplasia as described in the latest WHO classification. Traditionally, oral epithelial dysplasia is categorized using 3 grades: mild, moderate, and severe. In order to provide a more objective and reproducible assessment, a binary system has been proposed, dividing lesions into low risk and high risk on the basis of the number of dysplastic features identified on histologic examination. High-risk lesions must show at least 4 architectural features and 5 cytologic features, whereas low-risk lesions show fewer changes and may also include cases of epithelial hyperplasia that lack any dysplasia. And the same concept is depicted here. Despite the limitations, surgical biopsy and histopathologic grading of oral epithelial dysplasia remains the gold standard in assessing risk and determining the management of these patients. Most authors agree that the risk of progression increases with the grade of oral epithelial dysplasia. Indeed, significant correlations between these parameters have been identified in several studies. In particular, the binary system may have better prognostic value and superior reproducibility compared with the current, more widely used 3-scale grading system. For example, in the first study shown here, 80% of high-risk cases progress to cancer, compared with 15% of low-risk lesions. With further evaluation, it is believed that the binary system will become the standard grading scheme. This study further validated the reliability and prognostic usefulness of the binary system, which showed superior agreement between specialist pathologists compared to the WHO system with similar prognostic ability. Let's move on to the molecular changes and corresponding markers investigated in oral PMDs. Nowadays, it is common ground that the various types of cancer represent different diseases characterized by specific molecular alterations. A basic realization is that carcinogenesis relies on the accumulation of molecular alterations. These alterations accumulate in a precursor cell, leading to the eventual formation of a malignant cellular clone. This is the concept of monoclonality. Therefore, the Darwinian theory of natural selection and survival of the fittest does not apply only to species, but also to premalignant and malignant cells. And here we can see how the progressive addition of mutations leads to successive clonal expansion. This is a diagram showing how the concept of multi-step carcinogenesis translates into oral PMD and cancer. Please do not pay so much attention into the specific order of changes, since this is not always straightforward, but rather to the general concept of progressive transition from normal to cancer through microscopic phases of hyperplasia, dysplasia, and carcinoma in situ, which correspond to the successive accumulation of genetic and epigenetic changes. And here again, the same concept of accumulating genetic alterations that underlie and drive the phenotypic changes. The aforementioned genetic and epigenetic changes can be classified into several categories. In this seminal paper, Hanahan and Weinberg specified that development and progression of cancer entails changes that affect 6 fundamental cell functions called the hallmarks of cancer, including sustaining proliferative signaling, evading growth suppressors, activating invasion and metastasis, enabling replicative immortality, inducing angiogenesis, and resisting cell death. In a subsequent paper by the same authors, 2 more hallmarks were added, namely deregulating cellular energetics and avoiding immune destruction, along with 2 enabling characteristics, specifically genome instability and mutation and tumor-promoting inflammation. The importance of identifying and studying the molecular hallmarks of cancer lies in the fact that each one of them, despite of or because of its complexity, provides ample opportunities for discovering diagnostic and prognostic markers as well as potential targets for preventive and therapeutic interventions. Indeed, the progress of cell and molecular biology has advanced our knowledge of the molecular basis of oral cancer and has prompted the investigation of a number of biomarkers as possible prognosticators in oral PMDs. In this recent paper, my colleagues and I tried to summarize the current status of molecular markers that have been studied in association with development and progression of oral PMDs. In spite of its complexity, this is a simplified diagrammatic view of some of the major molecular events which have been suggested in the literature to participate in the development and progression of oral PMDs. Not all of them need to happen, Instead, various combinations of progressively accumulating molecular aberrations may occur, facilitating the transition from normal mucosa to oral premalignancy and eventually to oral cancer. Due to time limitations, it may not be practical to even mention all parameters studied so far. Instead, we can just briefly focus on two of the most promising areas of study, which are DNA ploidy status and loss of heterozygosity. Loss of heterozygosity is a form of allelic imbalance characterized by the loss of normal function of one allele of a gene, while the other allele is already inactivated. This may result in the absence of a functional tumor suppressor gene in the lost region. In general, when one allele of a tumor suppressor gene remains active, the gene may still exert its function. However, the second allele function may be lost through a number of genetic events. This is called loss of heterozygosity. Importantly, a number of studies have shown correlation between loss of heterozygosity at specific chromosomal loci harboring important tumor suppressor genes and risk of malignant transformation in oral PMDs. In these particular studies, the initial correlation of loss of heterozygosity at specific chromosomal loci with risk of malignant transformation was validated through a large prospective cohort of oral PMD patients, demonstrating that high-risk lesions, as identified by loss of heterozygosity using specific markers, carry a 65-fold increased risk of progression to malignancy. Another important parameter in oral preneoplasia is aneuploidy, which refers to abnormal number of chromosomes due to chromosomal instability. The percentage of aneuploidy in oral cancer has been estimated to be up to 90%. Similarly, aneuploidy has been detected in a significant percentage of oral PMDs and has been correlated with both degree of dysplasia and likelihood of malignant transformation. For example, in this study, a correlation between presence of aneuploidy and progression to cancer was shown, since the percentage of aneuploid cases was significantly higher among cases that progressed to malignancy. As shown in this recent meta-analysis, DNA aneuploidy may be one potentially useful biomarker for oral PMDs that has yet to be fully exploited, considering that all studies support a positive correlation between aneuploidy and severity of dysplasia as well as likelihood of malignant transformation. In summary, it can be said with a high degree of certainty that in future, special emphasis will be placed on molecular carcinogenesis with a focus on validating molecular prognostic and predictive indicators, ascertaining the risk of cancer, as well as identifying appropriate chemopreventive agents that will target specific molecules aiming at preventing malignant transformation. And this brings us to our next topic, which is the role of chemoprevention in oral PMDs. The term chemoprevention has been traditionally defined as the use of various substances with the potential to prevent, stop, or even reverse the progression of carcinogenesis. Several natural, synthetic, or chemical compounds have been used both locally or systemically with promising results. The definition and concept of chemoprevention has been modified over the years, so that recently it refers not only to the potential effects of these compounds on the multi-step process of carcinogenesis of epithelial cells alone, but also on the whole microenvironment, including the modulation of the immune response. The theoretical grounds of chemoprevention lie on the concepts of multifocal field and multi-step carcinogenesis. Taking into consideration the fact that genotypic and phenotypic alterations occur and progressively accumulate within a broader field, thus frequently resulting in multiple synchronous or metachronous lesions, chemoprevention seems as an ideal intervention aiming at arresting one or several of the steps, thus impeding or delaying the development of cancer. Accordingly, it has been proposed that the best target population for chemoprevention includes patients with excessive exposure to etiologic factors that contribute to an increased risk for developing oral cancer, patients with oral leukoplakia with high-risk features for progression, or post-operative oral cancer patients in order to prevent the development of a secondary tumor. Additionally, chemoprevention may be an option treatment for elderly patients who may have difficulties undergoing extensive surgery, as well as in cases of multifocal lesions such as proliferative verrucous leukoplakia that may be difficult to eradicate by surgical means. The list of chemopreventive agents used so far is long and includes both blocking agents, neutralizing the action of carcinogens, and suppressing agents, preventing the progression of the process of carcinogenesis. Several of these compounds can be seen here, including retinoids, COX-2 inhibitors, green tea extracts, and EGFR inhibitors. As you can see, the efforts on chemoprevention of oral PMDs go way back in the past, especially using retinoids with initially promising results, but with several side effects and frequent recurrences. Several other chemopreventive agents have been used in the context of oral premalignancy, as summarized in some recent review papers, as seen here, as well as here. Nonetheless, for almost all of these agents, and despite some initially encouraging results, several disadvantages have been noticed, including side effects even with low doses, as well as a large number of relapses after the end of treatment. So far, none of these approaches has proved to be consistently effective in order to avoid relapses or malignant transformation of oral potentially malignant disorders. To circumvent these problems, it has been proposed that ideally chemopreventive agents should target specific molecules that are known to play a key role in the process of carcinogenesis. In addition, more well-designed, randomized clinical trials of adequate size are needed, while an emphasis should be put on predictive molecular markers with a long-term goal of developing individualized approaches. In the last part of this presentation, I would like to share with you some of our studies, just as an example of the long process from basic research to potentially useful clinical applications. The focus of our studies has been to investigate the anti-tumor effects of NSAIDs and COX inhibitors on oral cancer. The anti-tumor effects of these drugs were studied before, both in vitro and in vivo, and were shown to be mediated through either COX-dependent or COX-independent mechanisms. The in vitro studies were performed in the School of Dentistry, University of Maryland, Baltimore. I was very fortunate to work under the mentorship of a brilliant investigator, competent pathologist, and very kind man, Dr. John Sock. We decided to study the anti-neoplastic effects of NSAID Sulidac, which were supported by clinical trials, animal studies, epidemiological data, and cell culture experiments in other types of cancer, However, they were unknown in oral cancer. This work has been quite fruitful, resulting in several publications written by me and my subsequent students and colleagues in Baltimore. Very briefly, we established that sulindac has significant dose and time-dependent cell growth inhibitory effects on human oral squamous cell carcinoma cells. In addition, it induced significant increases in apoptosis of these cells. More importantly, we were able to demonstrate that these effects were accompanied by significant decreases in activation and expression of the oncogenic molecule STAT3. STAT3 appears to have a key role in cell survival, motility, and tumorigenesis in several types of cancer, including head and neck cancer. In addition, the phosphorylated and activated form of STAT3 has been detected in oral paraneoplastic lesions, so that its inhibition may be a potentially important chemopreventive strategy. Constitutive STAT3 signaling in head and neck cancer has been linked to aberrant upstream signaling events such as JAK and EGFR signaling. Following tyrosine phosphorylation of STAT molecules, dimers are formed and upon translocation to the nucleus, they bind to promoter elements of specific target genes that encode for oncogenic proteins. Several STAT3 targeting strategies have been suggested in order to abrogate its oncogenic activity. Therefore, our observation that sulindac will inhibit activation and availability of STAT molecules was of importance. Furthermore, we were able to show that sulindac also downregulated the protein and mRNA expression of survivin, an anti-apoptotic molecule, which also was driven by STAT3 activity. Actually, these effects of sulindac on survivin were specific compared to other COX inhibitors. Indeed, the effects of sulindac on cell growth and apoptosis of oral cancer cells were quite similar with those induced by selective inhibition of survivin and were specific for sulindac compared to other COX inhibitors. Our major conclusions from the in vitro studies were that sulindac exerts a significant apoptosis-mediated growth inhibitory effect on oral cancer cells, which was mediated, at least partially, through downmodulation of the oncogenic molecules STAT3 and survivin. Based on these results from cell culture experiments, we proceeded with in vivo studies. In this paper, in which I had the chance to be a co-author along with the chair of this conference, Professor Chaisuparat, we used a xenograft model of head and neck squamous cell carcinoma, and we evaluated the effects of sulindac on tumor burden, STAT3 activation, and survivin expression. For these experiments, HEP2 squamous carcinoma cells were injected subcutaneously to nude mice. Following tumor formation, animals were treated with sulindac or left untreated as a control group. A significant decrease in tumor weight was noticed in sulindac treated compared to control animals. Further, decreased protein expression levels of activated STAT3 and survivin were seen in tumor tissues of sulindac treated versus control mice. From this work, we concluded that at the cellular level, the downregulation of STAT3 and survivin by sulindac was directly linked to its effects on cell proliferation, since forced expression of survivin and STAT3 reversed these effects. In the in vivo model, sulindac was able to significantly reduce tumor burden and to abrogate activation and overexpression of STAT3 and survivin. The last experiments I want to show you were performed in the University of Athens, Greece, and represent the continuation of our previous work in Maryland. In this study, we embarked upon exploring the anti-neoplastic effects of sulindac and its potential for chemoprevention and management of oral epithelial dysplasia and oral cancer, using this time the hamster's cheek pouch oral carcinogenesis model. Specifically, 30 serial golden hamsters were used, divided into 3 experimental groups and 2 control groups of 6 animals each. The experimental groups differed in terms of application of the carcinogen DMBA and sulindac administration, as shown in the table. It is interesting to mention that in this model, all animals are expected to develop oral premalignant lesions by the 9th week of the carcinogen application and oral cancer by the 14th week. Following animal sacrifice, the treated buccal pouches were examined macroscopically, histopathologically, and immunohistochemically. Macroscopically, it was noticed that animals in experimental groups receiving DMBA for 14 weeks developed tumors of varying number and size. Significant differences were recorded in total tumor volume among these groups, with the largest tumors in control animals, the smallest in group A2 receiving sulindac from the first week, and intermediate-sized tumors in group B receiving sulindac from the 9th week. Even more interestingly, microscopic examination revealed that invasive cancer developed in all animals in control group, while 1 animal in group B and 3 animals in group A2 did not show evidence of invasive cancer, but only developed epithelial dysplasia or carcinoma in situ. Also, the squamous cell carcinomas in animals receiving sulindac were more frequently well differentiated compared to the control group. Again, only 3 out of 6 animals receiving sulindac from the beginning of the carcinogen administration developed invasive cancer, the others developing carcinoma in situ in 2 cases and severe dysplasia in 1 case, as seen here. In addition, higher levels of immunohistochemical expression of the proliferation index Ki67 and the anti-apoptotic molecule sarvivin were noticed in the control group compared to sulindac-treated groups. As we can see in this diagram, sarvivin positivity was significantly lower in tumors developing in animals treated with sulindac compared to the control. All animals exposed to carcinogen for 9 weeks developed epithelial dysplasia, as expected. However, parallel administration of sulindac appeared to reduce the degree of epithelial dysplasia from severe to moderate in 5 out of 6 cases. Further, Ki67 and sarvivin immunohistochemical levels were higher in dysplasias of the control group compared to sulindac-treated group. Again, the percentage of sarvivin positivity in dysplasias developing in animals treated with sulindac was significantly lower. In conclusion, sulindac appears to exert anti-neoplastic effects in the hamster's cheek pouch oral carcinogenesis model. Specifically, sulindac was able to reduce the severity of epithelial dysplasia, to partially prevent oral squamous cell carcinoma formation, to reduce tumor growth, and to partially prevent the transformation of already established oral epithelial dysplasia to oral squamous cell carcinoma. These effects were at least in part attributed to sulindac's ability to reduce cell proliferation and to downregulate sarvivin expression. Based on all these previous results, we propose that the next logical step will be to design and perform a randomized controlled clinical trial aiming to investigate the effectiveness of sulindac administration in patients with oral premalignant lesions in correlation with its effects on specific critical oncogenic molecules such as STAT3 and sarvivin. Let us now reach our final conclusions. It is apparent that the various therapeutic approaches for oral PMDs frequently have only transient results. The main reason being that despite clinical or even microscopic remission, molecular alterations often remain. There is no doubt that there is a significant number of experimental findings shedding light into oral molecular carcinogenesis and resulting in an ever-expanding number of biomarkers and targets of potential value. However, the main problem is their application into clinical practice. Indeed, the current inconsistency of obtained results prohibits the use of biomarkers and molecular targets in a clinical setting. There may be several reasons for this inconsistency. First, the molecular heterogeneity. It is true that different oral potentially malignant disorders may have the same histologic subtype, but great variation in their molecular profile. Also, there are differences in methodology, use of different protocols, different reagents, and different methods for analysis. Furthermore, the differences in experimental design, especially in terms of patients and material selection, as well as length of follow-up, make it difficult to compare the various studies. But even more importantly, it should be emphasized that focusing on a limited number of genes and proteins, we may underestimate the complexity of the malignant transformation process. We may need to accept the fact that when we talk about individual patients, any single molecular biomarker may have limited capacity to reliably predict outcome, and any single molecular targeted therapy may have limited ability to uniformly prevent malignant transformation. But let us be optimistic. Despite the fact that evaluation of oral epithelial dysplasia remains the gold standard of malignant transformation risk assessment, it is an undeniable fact that our knowledge of the complex molecular mechanisms underlying oral malignant transformation has improved tremendously in recent decades. We were able to make significant correlations between specific molecular markers and risk of malignant transformation. And also, we were able to identify several potential targets for chemoprevention and therapy. This is my closing remark. The road ahead seems to demand an even better understanding of the critical molecular events heralding and driving oral PMD progression to malignancy. It is reasonable to expect that new methodological and technological advances will lead to the identification of more accurate markers for determining the risk of malignant transformation and better targets for chemopreventive and therapeutic applications in a more individualized manner. To achieve that, we all should try to create the necessary environment of international collaboration which will make feasible to perform well-designed, large, multi-center translational research studies. Thank you very much for your attention, or, as we say in Greek, ευχαριστώ πολύ.